Great. Welcome, everyone. It's really my pleasure to introduce Dr. Benedict Paten, coming to us from UCSC. So Dr. Paten studied neuroscience at University College London, and then went on to get his PhD in computational biology at Emble and Cambridge University, and then went over to UCSC to do his postdoc with David Hausler, and stayed on there to start his own group. And we're really excited to have Benedict here because he has played a really leading role in sort of this new revolution in using this idea called pangenomes, where instead of having a linear reference genome, you have really a collection of genomes that you would use as a reference. And I think this is a really cool idea, but it took a lot of hard work and a big group to kind of pull this off. And Benedict has really played a leading role in all of that. And it's something that really kind of has the power to transform a lot of the ways that we think about doing analyses that we've always done for the last 20 years using a linear reference. How can we start thinking about that in terms of pangenomes and the many different applications? So I'm really excited and have been, awaiting for Benedict's talk, and I will waste no more time and let him take it away.
Awesome. Does this work? Hopefully this works. Okay. It assumes as it works. Okay. Yeah. Thank you very much. Thank you for the kind introduction, Melissa. This was a little bit tricky. These slides are a little bit of a mess because there are a lot of different topics in this whole space to go about, and I kind of just fixed on a few. So there might be some discordance as I go through. I want to start off by just saying that my group, we're very pragmatic. We're interested in what allows us to get to, perfect genome inference, and we'll use whatever is necessary. And this pangenome is a sort of a tool in that toolkit to help us get there and help us think about how it all relates together. And at the end, I'm going to hopefully talk a little bit about some of the future thinking work we're doing about how we actually personalize the pangenome reference. And we have some nice results to show on that. Okay, so I'm going to start at the beginning. So, the human reference genome, right? Published, 2001. This was just a little bit before I, entered graduate school, and it really acts as kind of this cornerstone of human genomics. And by the way, the current version, for anybody who's not aware, is GRCH 38. So I'll probably quite often just use that language, but I mean the current human reference assembly, right? And the reason it's a cornerstone, and in some ways created that whole field, right? It's a proxy to a universal coordinate system, right? When we talk about genes and annotations and variants, we often, when we want to be precise, we use coordinates with respect to that reference. We talk about genes and so forth. I'm a PI of the GenCode project. All of our annotation is expressed on that reference, okay? And obviously, it's fun to think just how much cheaper DNA sequencing has gotten. We're all familiar with this idea, but just as a historical note, it took $3 billion in an act of Congress to get this first genome created. And yes, it's been 20 years since it was released, but it's still actually incomplete in that it was built originally to represent the euchromatic portions of the genome, so around about 90% of the genome, right? And those other bits, right, even in today's reference, even in GRCH 38, are largely missing, incomplete, simulated, right? So they're, in some senses, we don't, even in that current reference today, have a complete picture of what just one haploid human genome looks like. Right. So fast forward 20 years to 2022, my colleague Karen Rieger and Adam Philippi at NHGRI, and a whole bunch of us actually got together and completed the first haploid human genome. And if you want to understand the contribution here, so this is the cover from Science from that issue, and all the so these are obviously all of the chromosomes lined up. So if you like a sort of carrier type sort of racetrack, and all the things in red, those are pieces of the sequence that were filled in by our new reference. It's about 200 megabases of sequence that was resolved, right? It's about 6, depending on how you count, 6-ish% of a human genome that was added, right? So huge accomplishment. Really lots and lots of work coming from this that allows us to look at regions that we just didn't know, exactly what they looked like at a base level. But of course, one reference, right, doesn't contain the polymorphic sequence that is present collectively in all of us, okay? There are more than 100 megabases, probably about 150 megabases of commonly polymorphic euchromatic sequence that's missing from any single reference genome, right? Because by definition, it's segregating, it differs, right? These are the large structural variants that are present in our population. And no single reference can represent that, right? And because of that, we're missing maybe 3 to 4% of the sequence of any one genome when we compare it to GRCH38. And that results in what we call a reference bias, right? Which is an observational bias, right? So it's this idea of a street lamp effect. It's easy to see stuff that's right underneath the street lamp, the stuff that's in the reference. Much harder to see stuff that's in the, that's outside the space of illumination, right? The stuff that's in the dark corners, right? That's currently missing. So how do we go about completing this and making a reference that doesn't represent, serendipitously one individual, because the current reference is basically one individual. It's about 70% from an African-American admixed individual from Buffalo, New York, right? How do we move beyond that and get to a reference that truly represents everybody as closely as we can? So building on a work of lots of different people, about 5 or 6 years ago, a bunch of us got together with NHGRI and said, look, now is the time to think about really, yes, completing genomes, telomere to telomere, getting complete genomes, but also building a better representation of human diversity. Let's reboot the human reference and create a resource that has, hundreds of genomes, right? So we initially targeted, in our first round, 350, we're well on our way to this number, diverse humans, right? In an attempt to get basically all of the haplotypes that are common, that is present at least a 1% frequency, right, in the human population. But of course, in doing that, in sequencing hundreds of references, right, we need to think about a more complex reference structure that doesn't contain one, but contains many. And therefore, we need to create a bunch of new tools, right, to actually think about working with this more complex representation of diversity. And I'll get to some of that a little bit later. So, sorry, this slide's slightly out of date. So obviously earlier this year, we released the initial draft human pangenome reference. You can read about it. It's completely public. It's all on the internet. There are, of the order of 50 or so assemblies in there. And of course, in addition to the assemblies, as I'll talk about, we have alignments. And we also tried to demonstrate that we could do useful things with this reference, because there's not much point in doing this if we can't then show that it can have downstream impact. So we started to try to kind of catalyze the community by building tools and applications that I'll get a little bit to. Okay, so just in terms of that initial draft, we sampled actually from 1,000 genomes. So 1,000 genomes is very convenient for us because there are cell lines for all the samples from 1,000 genomes. So we could get high quality long read DNA for them. And they are they're consented for open data release. Although that's actually a question right now, but broadly they're consented for open data release. And 1,000 Genomes did a careful job of going and sampling a good selection, not a comprehensive selection, but a good selection of global populations from which we could sample diversity. So we picked our initial 50, and in fact, we picked our first 350 from 1,000 genomes. And the way we did that, it's an interesting question. How do you pick which samples should go into the reference? And we looked at a variety of approaches, but the simple way to understand this is that if you plot out a PCA plot of human diversity, you get this kind of V shape, okay? And we attempted essentially to pick samples under some constraints about trying to get samples that were high quality in terms of the cell line material, right? But we picked samples that tried to cover this kind of V shape, right? Now, it is important to emphasize that there are some populations and regions that are missing from 1,000 genomes. So, for example, there's really no representation right now of populations from the Middle East. And there are plenty of other populations, and particularly historically marginalized populations that are not present in 1,000 genomes. And in the long run, obviously, we are engaged in a deep conversation with various communities to try to eventually have samples that are either available for those communities to use or, in an ideal world, integrated into our pangenome. But anyway, this is where we started. So we had, samples of this particular makeup. And you'll note that, the majority of samples actually come from 1,000 genomes African samples. And that's because the majority of human diversity actually still exists in Africa, because obviously, the out of Africa bottleneck reduced a certain amount of diversity. So if you're actually interested in just trying to capture the most amount of population diversity, Africa is a rich resource. Okay, so what is powering the pangenome? Why was this possible? So I want to draw people's attention to this because I think folks have maybe not cottoned on to just how dramatic the changes have been. But obviously over the last 5 years, with the advent of PacBio hi-fi sequencing, with the ultra-long ONT and various other sequencing technologies, we now have technologies that for relatively low cost, we're talking about less than $10,000, right? And I think shortly less than $2,000 or $3,000, right? We can generate data that allows us to then assemble de novo, so without reference to any existing sample, a human genome at dramatically higher quality than anything that we could achieve in the previous 20 years, right? Each of the assemblies that, as I'll take you through, each of the assemblies that we sequence for the pangenome are dramatically better than that initial human reference, right? Despite costing just a tiny, tiny fraction. And that story has not finished, right? We are rapidly converging to a point where we can get diploid telomere to telomere genomes. See this latest paper from Adam Philippi's group on Verco, but actually there are other works from Heng Li's group and so forth, right? We are rapidly converging towards a world in which it is possible to assemble complete T-to-T genomes for, yes, just a few $1,000s. And if we weren't able to do this, then this project would not have had legs. We could not have put together the data that we have, and had the accuracy that I will show without that. So this is sort of the backdrop, the thing that enables us to do this. But much like large imputation panels like HapMap have been really useful for chip chip sequencing, right? Because they allow imputation. I think that these assemblies are going to allow imputation from short reads of all of the rich structural variation. So there's this direct translation of technology from the slightly more expensive through to the more commodity. Okay, so this actually now already out of date, but for the initial draft, we put together an assembly pipeline that was based on a big bake-off of all the contemporary tools at the time. And what was interesting actually about this bake-off is that it really established that it was no longer sufficient to do what we'd call sort of merged or haploid assembly, where you don't deconvolute the phases of the two, the parental contributions, right? That just doesn't work anymore. And it became very clear during that bake-off that the methods that were doing phase resolution were dramatically better in terms of quality from those that produce a sort of squashed assembly of the two things. So that was one, I think, big outcome. And the other was just that the hi-fi data, which was the new technology we had at the time, just allowed us to create vastly better assemblies than anything else. So anyway, I won't go through the details here, but we put together an assembly pipeline. This was all published this time last year, I guess. And as part of the effort, partly because a bunch of us are involved in the Anvil project, we tried to make all of the pipelines that I will talk about today, including this assembly pipeline, available for kind of button click reuse on Anvil. So you can go and look up on Dockstore for these pipelines, and they're relatively simple to run. So anybody can come and reproduce our assemblies if they have the input materials that they need. So in terms of, oh yeah, go ahead.
Weren't there additional IC and other platforms required at the raw assembly?
Yeah, so I skipped over. So actually for the initial draft, no, we just used trio sequencing. So we had Illumina for the parents. And obviously if you've got Illumina for the parents, you can pick out high quality k-mers that mark the heads, right? And therefore allow you, if you construct your initial overlap graph, now you do all your contig and so forth, you can then essentially label all your contigs with the trio information, and then that allows you to pull out a largely phased assembly. So the only inputs that we required for year one was hi-fi and that Illumina information. But since that time, because obviously people are really interested in being able to do this without having parental information, we've shown that, yeah, for sure, Heng's group and others have shown, actually my group too, have shown that it's possible to do this with Hi-C. Works great.
Does the current hi-fi ASM have a trio?
Yep.
As a standard?
There's a trio mode, there's a Hi-C mode. And now, the big new thing is there's also an ONT integration. I won't talk about this, but there's an ONT integration as well, which allows, that's really what's allowing us to now get through the last few hundred gaps and get to T-to-T. So it will be that combination of the ultra long and the hi-fi. Yeah, I could get really wonky about this. There's a lot of details. So in terms of contiguity, again, this is sort of already getting kind of out of date. But just to give you a sense of this, so this is an NGX plot. So if you look at this mid line here and you read it off, the point you have on the vertical axis represents the N50, the point at which 50% of the bases are in contigs of that size or longer. So if we take GRCH38 and split it wherever there's a contig break, then this is the line that we get. All of the purple lines represent the assemblies from the HPRC. These are all haploid, so they're the individual, they're at 90 or so, right? 90, well, nearly 100, I guess. And then this line represents kind of the theoretical maximum, which is the T to T genome, right? So you can see that these assemblies are highly contiguous. They're in the, I think the average was something like 40-ish megabases, something like that. Highly contiguous, but not quite perfect, which corresponds to a few hundred gaps that are left over, okay? And the remaining piece of this T to T effort now that we have ongoing is really just fixing 200 or so gaps, which is not actually that much sequence, but they turn out to be really tricky. Anyway, in terms of base accuracy, this is just showing you the QV quality value of the assemblies. They're about a QV of about Q50-ish. And that means that we have a base error about once, I think it was once every 200,000 bases, okay, on average. Right, that's across the whole genome. If you look in the sort of high confident regions, the genes and so forth, that accuracy is significantly higher. You're closer to 1 in a million. But it's sort of, the errors are dominated by certain regions of the genome that are just harder to assemble. And again, that is something that continues to increase. And I think our next round of the HPRC will be in the 60s, mid 60s. Our aim is to get to Q70. So that would be one error every 10 million bases and have less than 300-ish errors per genome. We think at that point, with current technology, we can't actually distinguish the errors. We get to the measurement error at the point that we have no way of knowing if we're any more accurate than that. So anyway, so this is where we are. So an error every 200,000 or so bases, which is pretty good. Also, just very briefly, this is no longer shocking to us, but at the time when we did these initial assemblies, it was really gratifying to see that assemblies, haploid assemblies that contain an X chromosome, come out at about 3.04 gigabases, right? Which is about 99-point-something% the length of CHM13, right? Which also has an X chromosome. And then the samples that don't have an X and have a Y are correspondingly shorter, because Y is substantially shorter chromosome. And of course, with the recent completion of the Y, now we are now rapidly moving to having complete Y chromosomes as well. So this is just to say that our assemblies are basically the full length, what we would expect a whole human genome to be. So one thing that we did really extensively was develop methods for assembly evaluation, because it's one thing to say, here's an assembly, trust me. It's another thing to say, here's an assembly and here's how I validated that this assembly is correct. Because any assembly is a hypothesis, right? It's a guess from your reads of what you think the genome is. And we did this a bunch of different ways. I'm just going to mention one, which was developed by a student in my group, Mo Benazri. And what essentially he did was develop it's a mapping-based approach whereby basically you map your high-quality reads. So in this case, this is hi-fi data, but we can also do it with ONT. So you map your hi-fi reads to the genome. And of course, in any region that is correctly assembled, those hi-fi reads should have a coverage distribution that looks, according to the binomial distribution, that looks approximately what you would expect. And if you have no reads, then if no reads can map to that location, then that's likely to be an error, right? If there are too few reads, then that's likely because you've artificially created a duplication of that sequence and the reads are kind of dispersing between the two copies. Or, and slightly more commonly, if you have a collapsed region, and this used to be really prevalent in short read assemblies, right, where you have under-assembled. You've essentially assembled one copy of the sequence when actually there's 2 or 3 or many that are present. So we fit essentially a simple Gaussian mixture model to the distribution, and we can show, I won't take you through all the evaluations, but we can show that about 99% of the assembled sequence is predicted to be, has reads that support the assembly. In some senses, it's like asking the question, what is the likelihood my assembly is correct given my data, right? Given my reads at this location. So this plot here is just showing you the fraction of each of the assemblies that falls into these different classes for all of our assemblies. And you can note the discontinuity in the plot here. So this plot would, if extended it all out, would go up to 3,000. But about 1% of our sequence falls into these categories that we are not sure about. Yeah, largely we have these false duplications still present in some of our sequences, but some other stuff too, right? So anyway, 99%, and of course, almost all, I won't take you through this, but almost all those errors actually fall out in the satellites. So satellite sequences are still the sequence where we are making mistakes. But even when we look at the satellites, about 95% of the sequence in our satellites is predicted to be correctly assembled. So all up, the upshot here is these assemblies are really crazy accurate relative to what we've had in the past. I could show you the same plots for assemblies that we're generating like just 2 or 3 years previous with earlier data, and they look wildly worse, like more than 10 times worse. Another thing that we did was develop a new, this is with the Ensemble folks, develop a new annotation pipeline for these assemblies. So essentially what we're doing is we're taking the GenCode annotation and then mapping that annotation to all of the assemblies, and then attempting to annotate the genes and doing a really careful job in the gene families to try and annotate the correct family members. That's really where the challenge is, right? There are many genes which look almost the same, and getting exactly the right annotation for the right gene can be challenging. The upshot is that our pipeline right now is not perfect, and it has some problems with certain gene family members. So for context, our complete and what we believe to be basically perfect CHM13 genome is shown in orange, and you can see that our HBRC assemblies are worse, but not a lot worse. Note that we're up in the 99s here. So about 99% of our protein coding genes that are in GenCode can be annotated. The non-coding genes are a little lower, and then the transcripts are slightly better. So anyway, the upshot here is that we have gene annotations for all of these sets, as I will walk you through, that we think are pretty high quality. I think is a really useful resource for people.
The discrepancy between CHM13 and the other genomes.
I think it's due to, I think it's kind of complicated. So there are certain regions that are underassembled, that particularly for the non-coding genes contain like fairly large arrays of genes, particularly on Y, right? You can see some of these dots down here. These correspond to samples with Y that are just which were not assembled correctly in this first round. So I think the discrepancy is largely due to assembly error at this point. As I say, there are some mind-numbingly difficult regions of the genome once you start getting into it that contain the most plastic genes. So we did a lot of work to try and find kind of validated gene duplications. We found over 1,000 copy number variable genes in our set of just 47 individuals. I think it corresponds to about 6% of human genes, protein coding genes, are actually copy number polymorphic in just 50 people, which is kind of mind-blowing, right? Like it's a huge amount of copy number diversity that's affecting a very large fraction of our protein coding genome. This plot shows you the number of samples for commonly polymorphic, copy number polymorphic genes, the number of samples, this is what the gray bar is showing, which are copy number polymorphic. So for example, the ZNF95 gene or GPRIN2, the vast majority of our samples have additional copies with respect to GRCH38. So the reference really is very insufficient for those samples because they just, they have extra genes and they're just not present at all in the genome. These blue dots represent the number, and the size is proportional to the number of samples, the number of additional copies. So for example, for ZNF95, many individuals have 4, 5 extra copies of that gene present in the population.
Is ZNF505 like a C2?
It is a crabting finger. I'm not sure.
Okay.
Yeah, you'd have to ask. So I should have said this was work that was performed by my grad Marina. She's now graduated, and Mark Chason's group. So if you have questions, definitely send them an email. In general, the crabting fingers are like
The most part.
yeah, right.
And you're confident that you're able to sort of differentiate.
Yeah, well, so the crabting fingers is the largest like expanded gene family in primates, right? But many of those copies are fairly old, actually. So most of them are like, yeah, it's not difficult. So things like the big crab cluster on chromosome 19, no problems, assembled perfectly. There are other loci which are much harder. The really recent, the ones that are most challenging are the seg dupes, where you have essentially the same sequence at 99.5% higher identity, right? More than one place in the genome. Those are the ones that like really cause us challenges. But yeah, I think most of those are pretty good. So this, just to illustrate, so it has been known since 1,000 genomes, just from like mapping short reads to the reference, that there was tremendous polymorphism. You can measure that just by doing copy number variation over genes, right? It was known that this polymorphism did exist, right? But it's only now, with the ability to actually assemble these things exactly, right, that we can actually see the exact structural makeup of these loci. So here, for example, I'm showing you this is a dot plot, so this is a view of the alignment comparing the two sequences, right? On this axis we're looking at GRCH38. On this axis we're looking at this just one of our samples that we picked, and you can see that GRCH38 has I guess where are we? Yeah, one copy of GPRIN2, but there are many copies of four, I guess, that are present in this particular individual. And of course, this is totally typical. This region is just like breaking and re-breaking, and you can see like inversion structures and so forth, right? The genome is in these regions just vastly more variable than the sort of like classical 1 in 1,000 prediction would have you believe. So one of the nice companion papers that came out with the pangenome work actually resolved, I guess, showed mechanism for a long-held observation. So it's been for a long time, people have known that the most common form of chromosomal abnormality in humans are so-called Robertson translocations. So Robertson translocations are where you take two acrocentric chromosomes, right, and you fuse them together, right? And it happens at about a frequency of about 1 in 1,000 births. Okay, so it's not rare at all, right? So why does that happen? Well, it turns out that when we looked in our assembly, this was initially actually a kind of quality control thing. Initially, we were looking for interchromosomal misjoins, right? And when we did, we found actually one interchromosomal misjoin that was a true assembly error, and we fixed it. It was not related to acrocentric chromosomes, right? But every single other predicted misjoin was between, and this is just a mapping-based analysis, was between the short arms of acrocentric chromosomes. So the short arms of acrocentric chromosomes, those are the places that they all localize together in the nucleolus, and they contain the rDNAs. It's the place where ribosomes are produced, right? So why the Robertson translocations? Well, it turns out that those sequences have these regions of like pseudo-homology, like long tracks of pseudo-homology where they are almost the same. But the only way that we could maintain almost the same sequence between different chromosomes, right, is if there is some process of gene conversion, i.e. recombination taking place between them. So what Eric's paper, this plot was produced by Heng, but what Eric's paper did was just dive into our assemblies, do a bunch of really careful work to show that actually there are these regions of homology along the acrocentric short arms in specific locations that allow for, presumably, some way for these sequences to be co-localized in the nucleus, right? But because of that, there is this process where every so often they recombine. And if they recombine in an inverted configuration, you get the Robertson translocation, because then in the inverted configuration, sorry, if to sort of do some DNA origami, you end up with like two of the long arms joined together, and then you've got this. Now you've reduced your chromosome number by 1. So anyway, so this is just a really nice demonstration that these assemblies could actually allow us to gain insight about what has been long held observation at the karyotype level, but without any real intuition about what the actual mechanism was. Another thing that I think is really neat about these assemblies, check the time here, is that we looked really heavily at some of the really complex loci. In particular, things like the big satellite arrays, in particular the centromeres. So the centromeres are the largest satellite arrays in our genome for a long time. So here is like a chromosome staining picture. We've known that centromere lengths vary by megabases between different alleles, okay? So this is actually two different alleles in one individual, right? Showing, this is just, showing the staining of the heterochromosome, right? They're dramatically different in length, right? But no one has ever been able to assemble them, let alone in lots of individuals. So we did that. We assembled and validated a bunch of these the higher order repeat arrays that are present. And we can show that, for example, here's chromosome 2. You can see that this is the length distribution. You can see that some people have commonly have an array that's like nearly 3 megabases in length, whereas some people have an array that's like less than 1 megabase. So why is that? It's because the centromere is resistant to recombination. There's no recombination within the centromere, right? And this is an important thing to do with the way chromosomes need to segregate, right? It would be dangerous if there was recombination. And as a result, because there's no recombination, they can evolve independently of one another. And so they're like little mini Y chromosomes, or they're sort of degenerate. They have a tree structure that relates them, and they are evolving on their own. And because they're very repetitive, they evolve by the gain and loss of repeat copies. There's this sort of like dramatic divergence where in humans, there's huge differences in the amount of varying sequence in these loci. So this is really nice, and it's really nice that we can start to put these things together. And I'll come back to centromeres in just a second. So, okay, so now, all right, I've talked about the assemblies. So what about the pangenome? Like, what about the other bits of it? So, okay, so in a cartoon picture, we take our people, we assemble our genomes. We obviously have 2 for each individual because they're phased, right? But because, of course, every genome looks pretty much like every other genome, we need to align them together. The alignment allows us to identify what is the same and what is different, and it is convenient to express that alignment in a graph, okay? The graph is nothing more than a convenient formalism that allows us to express any form of general sequence alignment. And so at this point, and normally I get this question from somebody in the audience, and they say, well, hold on a minute, why do I need this genome alignment? It seems really complicated. I don't want to deal with graphs. Like, what's the point? Like, I appreciate it's pretty from a theory point of view, but why can't I just pick the sample in your set that is closest to me? Let's say I'm British, right? I just pick the British reference genome and use that. I'll use that for my sample, for any British samples. And, okay, over here, this other population, I'll use their population reference. I want to disavow you of that notion. It doesn't work. It's a rubbish idea, okay? Okay, so this is not new information, but for a long time, we have known that, from 1,000 Genomes and HapMap, that common variants are mostly global. So they're present across all human populations. And that's because they predate by a long way all of our notions of common human migrations, right? They are typically about 1 million years old. That is long before humans were, out and about in the rest of the world, right? And actually, common variant frequencies, with some exceptions, right, because of bottlenecks, but mostly don't vary much between populations, okay? So, yeah, of course, there are exceptions. But if you look at things like FST, the vast majority of variants are actually shared and have relatively uniform variant frequencies across the human population, right? And if I look at any genome, right, I take your genome and I compare it to the reference, and I just label all of the alleles in it and say, okay, is this variant common, as in present at 1% frequency or higher? 96% plus of those alleles in any sample on the planet will be common, right? Almost all of the variants that you see in a genome are common, right? So they're old, shared, and they account for all the variants. Yes?
This is when you constructed a genotype matrix of the whole population. You haven't just sequenced one gene.
I need to be able to assign a frequency to any one variant. So I need the effect equivalent of nomad, right? But if I do that, right, and so I
extract all the alt alleles from a single genome.
You
ignore all the homozygous fragments.
Yeah, of course. Anything that's fixed, we don't care.
From a genome.
Yep.
The 96%
are present. Yes. This is not controversial. It's like known for a very long time. Yeah. That is not so. So the reason you're probably scratching your head is that obviously the vast majority of, if I aggregate all the variants together, say like I look at all the variants that are in nomad, the vast majority of them have frequency below 1%, right? But that is because there is an enormous tail of rare variants. Yeah, of course. I mean, across every human on the planet, you will find somewhere somebody has a variant at pretty much every site, right? But they're vanishingly rare, right? So the upshot of this sort of like the importance of common variation, right, is that if I take any 2 randomly selected genomes from any significant population, they'll differ by millions of variants. So if I picked me and the British reference genome, there will be millions of variants between me and the British reference genome. Because if you think about what population structure is, right, it's this like flowing of genes, right, of alleles across the population. We're always going to differ by millions of common variants. It's just, unless I pick, essentially my mom, right, then I am going to differ by millions of variants to that reference. And because of these facts, any genome that I pick, it will basically be dominated by those common variants. And so I will look essentially the same with respect to any reference that I pick. But in contrast, if I take a pangenome of just a few hundred individuals, and honestly, the dirty secret here is if I could have picked 100 Icelanders, right? Assuming, I don't know how many Icelanders we have in the room, but or any population, right? And I would find that if just give me a few hundred of those, and you will find that I have only a few tens of thousands of variants in my genome that are not present in that sample of Icelanders. And that, again, it's just because most of our variation is old and most of it is common, right? And most of it doesn't have anything to do with the way we currently think about human populations. Okay, so anyway, so this is just my argument. Forget about population-specific references, they're a bad idea. So we need genome alignments to express all of the relationships between assemblies. And again, people often come to me and say, oh, this alignment, this graph stuff, I don't know about it. It seems really CSE. I'm a biologist. I don't like it. I don't want anybody who, is not a developer to have to worry about this. I want you to understand that it is just a very convenient framework for us to think about data structures as a data structure, essentially, to think about algorithms for doing, genetics, right? And just to give you an intuition about this, basically our graphs have nodes and edges. The nodes in our representation, there are different ways of saying the same thing, but in this representation, the nodes have sequences, and then the edges represent the connectivity relationships between sequences. And then very importantly, in all of our conception here, yes, we have a graph, yes, it has a topology, but we store all of the haplotypes that flow through that graph, okay? And that's really important because linkage is important. And so we need to, there are obviously in any graph, almost an infinity of number of ways of traversing through it. Only a very small number of those paths will actually be biologically present, right? And that is why the paths are very important. Okay, so another sort of data structure question, again, I'm careful of the time here, right, is that, obviously as we, if we expand our reference from, GRCH38, roughly 3 billion bases, our set of 90 or so, 97 or whatever, haplotypes is 270 billion bases of DNA. So now have we, if we multiplied by 100 the size of the data that we need to ship around, if we want to do comparisons to a pangenome, the answer to that is obviously no, right? Because we can exploit huge amounts of compression from the fact that human genomes are all extremely similar, right? So turns out that if you take the raw sequences, yes, that's 270 gigabases of sequence or so, to gigabases roughly. You can compress that down, actually, I guess this is gigabytes, right? You can compress that down with gzip down to about 70 gigabytes. That's not a very efficient compression though. If you construct GFA, so GFA is essentially a representation of the graph that also includes these paths that flow through it. And it's increasingly becoming the sort of like simple format that everybody is using. You get about 45 gigabytes. That's okay, but it's kind of a lot, right? It's like the size of a fairly big BAM file. No one wants to have that shipped around all the time. You can compress that down to about 11 gigabytes with gzip, but actually, this is work from Yoni in my lab. If you're smart about the way in which the haplotypes are expressed, and we've created this format called GBSBC, you can read about it in bioinformatics, we can actually get this thing down to about 3 gigabytes, or roughly the same size as the original reference, maybe a factor of 3 bigger. And we think that further compression is possible, and further, the amount of compression grows super non-linearly. So once we can scale this to thousands, and we still only, we're still under 10 gigabytes of space. So storing loads of human genomes in this format is not a problem, and distributing them is not a problem. So anyway, an important footnote in all of this story. So building pangenomes is actually, when we started this, was it still is, an open question. And so in the project, we actually trialed, we had a sort of competition, and we trialed 3 different approaches. So one from Hangley, Minigraph, super nice tool, really fast, like everything from Hang. but it only represents structural variants. It's a way of building a coarse representation of a set of genomes that represents the large structural variants. And it has a lot of value, but it isn't for small variants. And then with Heng's lab, my group created, we sort of combined this cactus tool that we've worked on forever for producing genome alignments. We combined the two to essentially take the mini graph and then create a refined version of it that contains all the base level details, okay, right? And one thing to mention is that currently, although not for much longer, it omits the centromeric variation. So everything that's heterochromes in basically gets cut out of the graph. And then finally, Eric's group, so Eric, huge force in this space, has built this thing called PGGB that is lossless. It doesn't attempt to cut anything out. It also doesn't use any notion of a reference. So Heng's mini graph has a reference like structure to it. It doesn't have any notion of a starting reference and does everything all pairwise. And it's very nice, but it has some challenges because the graphs it creates are rather complicated and we can't currently map to them right now. But anyway, it's a really useful tool. And my point of showing you this is just to say that the problem of building one of these alignments is still an open problem, and we are converging towards better and better solutions. Very, very briefly, our pangenome contains, of the order of 20 million small variants broken up into SNPs, MNPs, and indels. In terms of structural variants, yeah, go ahead. Sorry, multi-nucleotide polymorphism, more than one base or closely spaced base. It's about 3%, is 3% of the rate of SNPs. So in terms of structural variants, there are about 70,000 sites that are structurally varying in our current pangenome. Okay, this is showing you it sort of three different ways, the three different approaches showing that they all kind of agree. And the coloring here, the light dark, is whether or not it's biallelic or multi-allelic. And then, like, if you look across our, this is the number of sites. Obviously, there's a lot of allos. There are a lot of mobile elements. These are all mobile elements, right? But and they're all mostly biallelic. If you look at the VNTRs and the STRs, right, they are generally not they are not biallelic. They typically have two, and in many of our VNTRs, we look at them, and almost every single haplotype has a slightly different allele, just showcasing just how fast the mutation rate is in those sites, right? And in terms of the number of bases that are affected in the genome, this is what this plot is showing. They're actually disproportionately, like nearly 50% of the bases of structurally varying bases in our genome are because of STRs and VNTRs, right? And then segdupes, they're a very small category of events, but a huge number of bases in our genomes, right? So anyway, so what I'm trying to say to you is that we sort of put together a fairly comprehensive picture of variation. The only class of variant that is really missing from our current lexicon are the inversions, and that's just because the inversions are quite difficult to get right with our current sequencing technology. There are some sequencing technologies like StrandSeq that allow us to get inversions right, and it's definitely a work in progress, but we didn't feel confident enough about them to really include them in this analysis. I'll skip over this and just say that our variants are all very highly accurate and concordant with existing variant sets. And here, I just want to illustrate, most of the pangenome is a boring structure. It just looks like left to right, matrix-like, traditional multiple sequence alignment with the occasional step, right? But there are about 600 loci in the human genome in our current set that are like large and structurally varying. So here I'm showing you, HLA-A. So this is part of the MHC. It's obviously important for antigen presentation. So it's evolving very, very quickly. And it does not, there is no linear path through this, as I'll illustrate, that captures all of the different sequence variation. So this is a sort of like, if you imagine taking our whole pangenome, which contains hundreds of millions of nodes, zooming right into this little piece, little fold, this is what this loci looks like. You can see, so here's HLA-A, and there's all these other homologs of HLA-A, paralogs, I should say, right? And one interesting thing is that it's been known for a long time that there was this HLA-Y allele, and that it was common in the population, but nobody knew exactly how it was structured, what its composition was. But with the pangenome, we can reveal that. So here's that same graph, and then here over on the right, so the genes here, by the way, I should have said, are colored, these little colored ribbons. But in this picture, the red to blue represents the path of one particular haplotype. So GRCH38 does not contain HLA-Y. So you can see that it kind of does this, right? It skips that whole arc, right? But then there's this other haplotype, which does contain HLA-Y. And it's about, nearly a third of haplotypes, between a quarter and a third, right? And it does contain HLA-Y and it skips this little sequence and so forth, right? And there is just tremendous variation in the population around these like major structurally varying alleles, right? So all I want to say, this is just one of, if you like, 600 examples, right? But there are these loci that just do not fit our classical matrix view of genetics, right? Where whole large segments, sometimes up to megabases, are gained and lost, right? And commonly variable in the population. So this is all stuff that has been published, and I guess I felt like I needed to tell you about. But now I want to talk about a couple of, like, in the time I have left, a couple of research topics. So one thing in the first round of the HPRC is that we really ignored heterochromatin, right? So heterochromatin, like roughly 10% of our genome. It's the stuff that comprises the satellites, the centromeres, the telomeres, the acrocentric short arms, right? This is all this like really hard, really repetitive stuff that's present. And our current methods just basically don't do a good job of it. So both the minigraph and minigraph cactus, right, they just clip it out. Like we're just like, I don't know what to do with the sequence. So when we find large portions of heterochromatin, it basically won't align because our aligners don't know what to do with it, and we just remove it. And then Eric's tool tries to maintain the heterochromatin, but when he does that, he can't actually align it. So we end up with a graph that has 5 billion bases of extra heterochromatin that's just floating around, and it's not a reasonable alignment in any way. Okay, so this is, if you will, the very last frontier, the last place that's completely unexplored, the last wilderness in our genome that we don't really have a good way of like mapping and thinking about and comparing. So, I think an open challenge right now is just how do we take this sequence and align it so that we could actually produce reasonable maps of the variation in this sequence. It's not good enough for us to just say, here be dragons, let's forget about this, right? Because there are chromosomal segregation disorders associated with centromeres. There's, obviously, the telomeres are important for a whole bunch of classes. We need to be able to assay this variation, right? And it's not good enough for us to just say, no, we're giving up. So actually crediting Pavel Pevsner, obviously, and Andre, who I think recently graduated from his group. So they came up with this really nice method for trying to align together centromere arrays. I think it's called, uni-aligner. And the idea of it is quite simple to understand. So if I here's a dot plot showing you, so again, it's like a 2D view of alignment of two sequences. So all the dark things represent matches. And you can see that this is a tremendously repetitive sequence, right? With much self-similarity between these two different sequences, closely related sequences, okay? Right? So that's all the matches. But if instead I show you exactly the same dot plot, but now all of those matches, I've changed their intensity depending on how unique they are. So if they're very unique, I've given them a dark color, and if they're like the same as lots of other copies, we've allowed the shading to be much lighter. And you can see that suddenly, whereas this is pretty difficult to determine what the alignment is, this looks much more like a conventional dynamic programming problem, right? Suddenly, when we think about it not from a similarity point of view, right? Because classically, all alignment algorithms are just similarity driven. They're looking for stuff. Is it the same? Okay, good. That gets a good score. If it's different, it doesn't, right? But if we instead think about it from a uniqueness point of view, then suddenly we can actually start to see the breadcrumbs that would allow us to create a reasonable alignment for at least much of this sequence. So when we do this, again, crediting Pavel's work, and then we're building on this, we can actually build alignments through these sequences that are otherwise just impossible to align with our current similarity approaches. And so this again, crediting Pavel's work, but this is an alignment of an X. What Jordan in my group, as postdoc, this incredibly talented postdoc, has done is then taken Pavel and Andre's framework and generalized it to not just pairwise, because Pavel and Andre's thing is just a pairwise thing. I generalized it to the problem of n dimensions, so how to create a multiple sequence alignment from the sequence. And there's a whole ton of very exotic data structures and algorithms that are being used here. But for the first time, we are able to build alignments. So this is chromosome 12. This is an alignment of 38 different haplotypes through chromosome 12. Chromosome 12, again, is just almost impossible sequence is just incredibly, incredibly difficult, right? And this represents the very first multiple sequence alignment of this array. And in fact, we're now doing this across all the different arrays. And so we think that there really genuinely is hope. I know this looks really complicated, but actually the structure is actually a DAG. And there is this vertex will represent, or this part represents the in, this represents the out. And it turns out that yes, there's a whole bunch of stuff to do with the way in which we choose to align and choose not to align, but we believe we can produce a reasonable alignment of this sequence that is going to allow us to do genetics on this sequence. We're going to actually be able to catalog individual variants and say, oh, yes, you have this extra copy and this associates with this particular chromosomal segregation disorder, etc. Aligning two genomes. Aligning reads is absolutely doable, but again, if you want to align it to a pangenome structure, so different topic, but short reads very, very difficult, but long reads, yeah, we think we can align into the structure. Okay, so I'm running out of time. So we've done a lot of work on applications of this thing. I will skip through this. So one tool that we built in my lab is Giraffe, which is this fast short read mapper that tries to basically make everything into an exact matching problem. And it kind of relies on the fact that you would think that alignment should get slower because you're aligning to more and more sequence. But actually, if your pangenome contains all the variants that are in your reads, your matching problem gets easier because your reads should look more and more like the stuff in the pangenome. So it turns out that we can make this really, really fast. And then working with, my group has a long-running collaboration with Google Health team. We developed a pipeline using Giraffe and then deep variant. And actually, in this is an interesting, we're mapping into the pangenome, but because deep variant doesn't understand the pangenome yet, we then project all those mappings back onto the reference. We gain because we're mapping into the pangenome, and therefore the reads end up more accurately placed. But at the end, we just project back onto GRCH38 for the purposes of variant calling, and we can show that improves our results. So the upshot is that the results are great. Extremely, extremely, extremely good. This is precision recall plot up and to the right. It's good. You can read about the details in the paper. I think we reduced the errors by about 30-ish% versus the remaining errors, which doesn't sound like much, but we're still talking about a few tens of thousands of variants that are now no longer wrong. I think the big near-term killer application of the pangenome will be in typing structural variations. And again, there isn't time to talk about this in any detail, but Tobias's group, Jana, developed this tool called Pangini, which kind of does a sort of imputation task on the haplotypes, but for structural variants. And we can show that compared to, so this is the number of structural variant alleles that you're typing per sample, okay? Now, the average human genome contains about 30,000 structural variants. If you use contemporary short read methods mapped against GRCH38, you can type about a third of them, maybe 9,000 structural variants with confidence. Okay, now I know that there are some specialized methods with certain types of sequence there where we can do a little better, but in general, there are many, 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 many structural variants that we just are kind of hopeless with the current reference. But with the pangenome, we actually double the number of structural variants that we can type. So we're going from like poor minority to at least a majority of the structural variants that we can type with the pangenome. And I think that's a big boost, and we're nowhere near diminishing returns on this yet. We think we'll be able to get to a point where with short reads, we'll be able to type the very large majority of structural variants in the genome. And then the very last thing that I wanted to talk about was this idea of where we're going. So most of the rare variation that we're going to add to the pangenome over the next few years is actually going to be absent from a given sample that's under study, because I told you about why everything's common, right? And that rare variation, it kind of acts like noise if you're naive about it. So if you just mapped the topology of the graph, if you just keep adding variation, then eventually you've added a variant at every SNP, at every base, and it just looks like this impossible structure, right? So how do you get around this, right? And just to illustrate this, right? Like, so here is a sequence tube map. Reads are in red here. Consider this purple haplotype that contains this apparently relatively rare G, okay? The other haplotypes, sorry for people who are not familiar with this view, the other haplotypes are these gray things. This G is very unlikely to be present in my sample at this location. right? And if you imagine this across the whole genome, this problem actually becomes kind of significant, right? As we grow. So how do we get around this? Well, the dirty secret of the pipelines that we have created to date is that what we do essentially is take our big, beautiful pangenome with its, extra millions of rare variants, and we just throw them away. We just say, okay, anything that has an allele frequency of less than, you can see over here, 1%, 10%, depending on what you're dealing with, right? We'll just throw it away, and we'll create a smaller subgraph of just the really common stuff, and that will be our substrate for mapping. But of course, that's a waste, right? Because any sample contains tens of thousands of those rarer variants, right? And we would really like to use them in our analysis. So how do we get around that? Well, hopefully it's given away by the title here. We think that the solution to this is really a personalization of the pangenome. So we imagine in the near future, as I'll show you, that if you take, initially just take k-mers from the sample that you are mapping to the pangenome, and you use them to just color the haplotypes in the pangenome. And then at every location, right, you pick haplotypes, so a subset of the haplotypes that match your sample, okay? We have two modes of this. Either at the location you can pick the M-best haplotypes, or you can pick the M-best, and then from that you can optimally pick, the diploid best combination, okay? And then, so it's like doing a local ancestry inference across the whole genome. At every location, you're picking the haplotypes that are closely related. And then right now, we just allow free recombination between those haplotypes to create like a sort of synthetic subset of the pangenome. And this is it's a subgraph, if you will, with a consistent set of haplotypes that flow through it. And the data structures and algorithms on this are truly quite beautiful. And you should see Yoni's work on this. So just to illustrate, so we can map to this subgraph. So this is the mapping time for mapping a sample to GRCH38 with BWA-MEM, right? It's from Heng, right? So it's about 2 hours on a 32 core node, right? And without the pangenome sampling, we're at about just over half the time because it's really fast, right? But using this pangenome sampling, I'm just saying we can achieve, runtimes that are a little bit longer, but still very, very doable, certainly faster than mapping to a linear reference. These are all short reads, by the way, I should say. These are NovaSeq reads, right? So really, really fast mapping to a personalized pangenome. And this includes the whole pre-processing step to like build that personalized substructure. So it's really, really practical. And just to illustrate, so this is the orange dots here represent the total number of errors that are being made by a short read pipeline for genotyping. And you can see that GATK is rubbish, right? But we've known that for a long time. Here's a deep variant against the linear reference. And then here's our personalized pangenome, right? It has I think it's about 45% fewer errors versus, so we're just picking the best pair of sequences. We're picking a diploid personalized graph for our sample to map to versus mapping either to a filtered graph or mapping to the whole pangenome. Slightly better than the filtered graph. And then just finally, if we do this for structural variants, again, so here are the current F1 scores for genome in a bottle, I think the 0.6 structural variant benchmark for HG002, and you will see that they are terrible. So all of these methods, DELI, MANTA, Scalpel, these are all, they're all state-of-the-art methods, but they are rubbish, right? Like this is a terrible F score, right? Dominated by just having absolutely appalling precision. They do not construct SVs accurately. Okay. Now, in contrast, long reads they're like killing it, right? Like their performance is great. This is not surprising. Their reads are like 100 times longer, right? Okay. But with our diploid mode, the pangenome, right, we're actually starting to approach the performance of long reads. Like look at the delta between that performance and like, what's Meta SV. That's apparently the best short read SV typer from the currently reported genome in a bottle set, right? So we're, approaching long read quality, but for short reads, right? Using just the current pangenome with just 50 samples in it, right? Now, our next round will have hundreds of samples in it. And as we scale, we see, we are not close to diminishing returns. We believe it's possible for our short read methods using this personalization approach to essentially achieve the same thing as we do with long reads. Okay, so I just want to summarize because I know I'm out of time. So I think the reason I'm involved in this project, the reason I've been thinking about this for, 10 years, right, is that I think it's really important that our reference and everything that we do in genomics tries to equitably represent everybody, right? Right. It doesn't work for us, for healthcare, for all the things that we want to do, if we privilege one group over another, right? So to do that, of course, we're trying to build a pangenome, and the draft that we've released thus far adds, of the order of 120 megabases of euchromatic sequence, many gigabases of additional heterochromatic sequence. Billions, hundreds of billions of base pairs of assembled sequence. 1,500 or so gene duplications, right? Millions of small variants. And this is by far the biggest library of high quality structural variants that exists right now. And I know that it's going to take, I keep telling people, it will take 10 years for this thing to actually become broadly adopted, because everything takes longer than you think, right? But in the long run, I think our job right now is to demonstrate that there are compelling advantages to doing this, because there's no point moving if we can't demonstrate that, right? And I think that we are on that path towards being able to first in research and then ultimately in the clinic improve genome inference, so that everybody can benefit from the knowledge of all variants across the genome in a better way. And just with that, I just want to say thank you to a lot of people in my group. These are all folks on the beach in Santa Cruz, folks at Google Health who have been really, really involved. And then more importantly, I guess, to just thank a lot of the people who are in the HPRC. This is from our meeting last year. Thanks. Yeah, sorry for going over time.
We're not in Kansas anymore. So on that slide that showed the difference between short read SVG and typos, like
Yep.
versus long read versus BG. I mean, BG is amazing, really good. But I had a detailed question this week. How much of typically if you just do SV calls with any random stipples 2 or something like that, 60% of the calls you get will be in Canopy.
Yep.
So are you including those in your genotyping layer?
Yeah, and actually that, yes, and it's fair to say that genome in a bottle benchmark is super biased towards the tandem repeats. It really doesn't. Right now, I think that benchmark contains like 10,000 structural variants.
So you wouldn't use MANTA to call SPs in Canopy?
No, this is true.
That's not what it's designed for. So you have to combine it with another TR gene.
Yeah, right. So that's fair, right? So it's definitely possible to couple, like the performance of deletion accuracy is much better than it is for insertions, right? Obviously, and obviously for longer, there are specialized methods for VNTRs. I think the advantage the pangenome has in general is that it kind of unifies all of that together. There's no need to subclass these together, because they're all just classes of sequence that looks the same once they're all present in the pangenome. And so I would appeal to I think you're right. Like, I think maybe this is unfair in the sense that, like, we just took the best, according to Genome in a Bottle, the best performing from their benchmark and put it on this slide, right? But you're right that we could definitely make a case that we're being unfair because we're assaying across all forms of structural variants, and some of them are designed for specific subclasses. But I think, we looked really carefully at the 1,000 genomes SV sets, right? And they are still only, and this includes, they did a lot of work to run a bunch of different consensus callers. They're still only in their confidence set calling of the order of 10,000 SVs, right? Where we know from any long read study, and yeah, run your snipples too, that you're going to get 25,000 out. That's, I mean, from an association point of view, that sucks, right? Like, to be missing the large majority is no good. So yeah, I take the point, but I think I would try to argue is that I think we can get to a point where we can really do this comprehensively. Yeah, and also I also didn't want to rag on those, but I know how hard it is. It's a problem. We've done work on that. It's really tough.
I have one more, but I'll let somebody else ask a question.
I have a little bit of question.
Okay.
I have also put there.
Yeah.
Okay, I'll ask one more, I guess,
Sure.
technical question. So you showed this like a new expression technique that you're
Mhm.
using that GBSBC. And I'm wondering how that compares to some of these, like ODGI has like an index structure that they build. I think there's like a GSAB thing that we've looked at, and like, how does this compare to the
I mean, that's a good question. So, yeah, and there's also just to complete that set. So Heng and forget the name of the investigator in Europe have been working on just like a general sequence compression thing, which is just like take all the sequence, don't think about its alignment, how much can you compress it? So I think it's good that we are exploring a diversity of options at this stage. I think the advantage that GBZ has in this space, I think it is the smallest. I think it ends up being the smallest at this point. The advantage is that it has is that we can make it into, because it's referenced across the pangenome, it can be indexed. So you can pull out small subgraphs really, really rapidly. I don't know about the ODGI tool, about how well that works. Eric is sort of building his own sort of infrastructure over on the right. Yeah, it's a fair question, but I think competition is a good thing. So yeah, so I'd love your feedback on that. And yeah, part of this whole, I should just say that the pangenome group has been a really tight group over the last 2 or 3 years, as we've kind of had our heads down trying to get some stuff done. But I'm hoping with this next round, as the number of investigators grows, that we'll have more people coming in and doing things like this. We will not succeed as a community if we try to hold on and like we're doing everything. Like that doesn't work. Like it has to be like a whole bunch of people adopt this stuff, and it becomes just like the bread and butter, and becomes really boring after a while. Like I was saying this to you earlier. Ultimately, the idea is that this thing will disappear into the background and just become, oh, of course, it's a black box. Most of the time you don't have to think about it, but of course it's there, right? There's a question behind you as well. Yeah. Yeah.
No, don't take me.
So far, yeah, I'm not.
Yep.
And even then,
the answer in general, yes. We're going to put a big effort into modularizing a lot of the like libraries that we created. You have to, like in all these projects, like in any like academic project, you start off by throwing a ton of mud against the wall. You make a bunch of messy libraries. It's all grad student code. It doesn't, it works to an approximation, it works, right? But then you have to get to a point where you then, the Hengleys of this world have to come along and package it up and make it super nice. And then, oh, everyone can use it, right? It works, right? So we're definitely thinking a lot about how to take VG, which I don't know how many tens of thousands of lines of code it has in it now, and modularizing it into smaller pieces built around specific tasks. In terms of the specifics around GFA and access and so forth, I think the answer right now is no, it doesn't work well enough. Like, for example, the index support that I was alluding to earlier is still a sort of prototype feature right now that we haven't released to anybody yet. So I think, yes, I think maybe I should be careful when I say that. I mean, we're definitely at a phase where early adopters, people who want to build interesting tools, can come in, and we have stuff that works, but I don't think it's as polished as it should be. And obviously it's going to take a few years to get there. For those of us who are old enough to remember like the initial round of SAM tools and so forth, there was a ton of mess there too. And it's going to be the same thing. Sure.
I had a small clarification. Right now, VG is using it's not using.
So VG is our toolkit. As I say, it's like big bucket of a lot of stuff. It contains mappers that will short read mappers, and we now have a long read mapper that we're nearly done with. It contains index formats and structures, and it contains a whole ton of utilities for messing around with the GFA files for a bunch of things. It also contains RNA and expression quantification. So it is a way too big project that has kind of grown out of control. And our intent is to like turn it into little bits.
Part B of my question. In clinical, majority of the clinically reportable grants have looked.
Yep.
If you were going to apply this clinically, then you obviously have to call rare variants that are probably not in the pangenome.
Yep.
How would you do it?
Yeah, so that's a great question. So I'd start by saying like having the haplotype background on which the variant occurs is generally really helpful. So we can show that de novos, oh, sorry, variants that are not present in the pangenome, we can call more accurately than you can call using the we've done this analysis versus just using the standard reference. So it's not like we're like depleting our accuracy for calling stuff that's not present. And the reason that we get an advantage is just because, okay, that variant might not be in there, but the actual haplotype on which the background on which it occurs and the variants that are around it, they are, right? And so you capture that. So yeah, it's a point that's well taken. I think one of the big things that's going to be useful for the pangenome actually as a use case is just sorting out what is like vanilla, the same, it's present in lots of people versus what is new and rare, right? And if you can reduce the size of what's new and rare to just a few thousand from from millions, then you've made your problem way, way better. And like I say, an average person compared to a pangenome, we're talking only about 10,000 variants that are not present versus 5 million.
Logistically, in terms of actually calling them a variant, I think I can understand. If you have an assembled gene, VG has no problem incorporating new loops in the graph, right?
Yeah, no, but you map your reads. So we definitely think of the pangenome as just like a mapping substrate. So you just map all your reads against the pangenome, and then this is work that's ongoing.
If you have something for which there is no part of the graph, how does that happen?
So for SNPs, it's just the same as calling as you would against a linear reference for a variant that's not present, right? And this is very unpublished work at this point. We have this big collaboration with the Google group right now on building transformer models for essentially making it possible to essentially just walk along. You've mapped your reads, these paths in the pangenome, you infer a diploid genome, and you walk along each haplotype, and all you're doing is just predicting the delta from those sequences for every, and we can show that we can call the differences very effectively that way. And that's a way of sort of creating a unified way of doing variant calling that works for all classes of variants.
Does it require adding little layers of like snipples? Like do you have to have like a little add-on SV caller that sort of like polishes out what's going on in the optimal? So you have an optimal path.
Yeah.
Through the graph, but it's not perfect.
We don't know that at that level of detail we don't know yet. But those are so rare. I want to just emphasize those are so rare compared to the vast majority of those things that are missing are SNPs and small indels, and those are easy. But yes, you're talking about maybe a few tens to hundreds of the really. Yeah, that's the final really hard part of the process. So
that's also where some of the most interesting pathogenic structural variants.
Yeah, for sure. Yeah, and I tend to think that the de novo assembly will be good for that. Yeah. So, thank you.